Hi guys, in this part I want to talk about tubular reabsorption and secretion. These two processes happen after the first process in the physiology of kidney, which we call it filtration. The volume of fluid which enters the proximal convoluted tubule after filtration is greater than the total volume of the plasma in blood because the glomerular filtration rate is normally so high. Obviously, some of this fluid must be returned back to the bloodstream, which we call it reabsorption or tubular reabsorption, the return of most filtered water and the solutes like glucose, amino acid, and ion. Normally, about 99% of filtered water become reabsorbed. And after that, we have the third mechanism, which we call it secretion. The secretion helps the managing of pH and help our body to get rid of toxins and all foreign substances. Before I talk about this table, I want to tell you that the epithelial cells which are located in the renal tubule and renal duct carry out reabsorption. And the most important part is proximal convoluted tubule, which can make the largest contribution for doing reabsorption. Reabsorption can take place by active and passive process. Different ions like sodium, potassium, calcium, chloride, bicarbonate, and phosphate, amino acids, and glucose become reabsorbed in our nephrons. Peptide and proteins are reabsorbed by pinocytosis. You know that the proteins and peptides are large particles. They cannot pass through this phospholipid bilayer of the apical border. So by the next process, by the other process, which we call it vesicular transport or pinocytosis, they can easily come to the cells, and after that from the cells they can reabsorb into our bloodstream. Once fluid passes through the proximal convoluted tubule, the cells which are located in the distal portion of the renal tubule can fine-tune the reabsorption process and maintain homeostatic balances for water and the other ions. In this table, you can see the plasma filtration and the urine composition. Here you see that the amount of water in our plasma is about 3 liters. Each day, 180 liters of water become filtered. From this amount, 178 to 179 liters become reabsorbed to our bloodstream, and only 1 to 2 liters of blood is present in our urine. The amount of protein in the plasma is about 200 grams. 2 grams become filtered. You know that the proteins are large and they cannot pass through the filtration membrane. And only we have about 2 grams of protein each day which become filtered. From this amount, 1.9 grams become reabsorbed, and only we can see 0.1 gram or no protein in the urine, and it's normal situation. The amount of glucose in our plasma is about 3 grams. 162 grams of glucose is filtered in our kidneys each day, and all of them should become reabsorbed, and we don't have any glucose in the urine. The amount of urea in our plasma is about 1 gram. And during the day, 54 grams of urea become filtered, 24 grams become reabsorbed, and 30 grams are present in the urine. And for the creatinine, we have 0.03 grams each day, 1.6 grams become filtered, nothing become reabsorbed, and all of them can be seen in the urine. So you see that the different material have a different destiny in our kidney. Some of them become fully reabsorbed, some of them don't have reabsorption, and some of them have both reabsorption and secretion. The third basic function which we have in our nephron and collecting duct is called tubular secretion, the return of material from the blood and tubule cells into the glomerular filtrate. Secreted substances include hydrogen, potassium, ammonium ions, creatinine, and certain drugs like penicillin. The secretion has two important outcomes. First, the secretion of hydrogen ions help control the blood pH, and second, the secretion of other substances helps eliminate them from our body. As a result of glomerular filtration and tubular secretion, certain substances pass from the blood into the urine and may be detected by urinolysis. It's specifically important to test athletes for the presence of some doping drugs or performance-enhancing drugs like anabolic steroid, plasma expanders, erythropoietin, HCG, HGH, and amphetamine in their urine. The urine test can also be used to detect the presence of alcohol and some illegal drugs like marijuana, cocaine, and heroin in the in the urine. We have two different routes for reabsorption. A substance being reabsorbed from the fluid in the renal tubule lumen can take one of two routes before entering the peritubular capillary. One of them happens between adjacent cells, which we call it paracellular reabsorption, passive fluid leakage between cells, and it's about 50% of reabsorption. And the other process happens through an individual tubular cell, which we call it transcellular reabsorption. And the other 50% of reabsorption take place by this method. In this picture, you can see the apical border of the cells. This is the proximal convoluted tubule, and you have many microvilli or brush border here. And the other border, which you can see, is called basolateral border. So here we have one apical border and three basolateral border, which we have which we have for each cell. Here you can see that sometimes the sodium ion can pass between the cells. You know that between two adjacent cells we have intercellular junction, but sometimes our intercellular junction become leaky and it helps some material to pass. This type of movement is called paracellular reabsorption. The other type of movement is called transcellular reabsorption. In transcellular reabsorption, a substance can pass from the fluid into the tubular cell through the apical membrane of the tubule cell across the cytosol, and then it goes out to the interstitial fluid through the basolateral membrane. So these are two different processes which we have for reabsorption. You need to know that the reabsorption have different mechanisms. When the tubule cells transport solutes out or into the tubular fluid, they move specific substances in one direction only. Various carrier proteins are present in the apical and basolateral membrane to help this transport movement. Reabsorption of sodium ion by the renal tubule is especially important because of the large number of sodium ion which pass through the filtration membrane. But sometimes, besides the diffusion or the facilitated transport or the facilitated transport with carrier or channel protein, we have the other mechanism like primary active transport and secondary active transport. In the primary active transport, which we have it for the sodium potassium pump, we use one ATP, and by using this ATP, we can move 3 sodium ion out of the cell and 2 potassium ion into the cell. At rest, about 6% of our total body ATP is used for this pump. And the other type of transport is called secondary active transport. In the secondary active transport, the energy stored in one ion electrochemical gradient can help movement of one particle across the membrane. Secondary active transport couples the movement of one substance downhill along its electrochemical gradient to the movement of second substance uphill against its electrochemical gradient. And we have two types of secondary active transport. One of them is called symport and the other one is called antiport. We have some proteins which are called symporters. They are membrane proteins which help two or transported substances to move in one direction across the membrane. And the antiporters are the proteins in the plasma membrane which move two or more transported substances in opposite direction in the membrane. And in this slide, you can see the symporter. This is the symporter, and it can help co-transport. Co means in the same direction, and you see that these two materials, both of them can move in the same direction. And the other type of movement or a secondary active transport is called countertransport. Here you can see antiporter, the protein which helps the material to move in two different directions. One of them can come to the cell and the other one can go out of the cell. All water reabsorption occur by osmosis. You know that osmosis passively moves water from an area of lower solute concentration to an area of higher solute concentration. Therefore, solute reabsorption drives water reabsorption. When we have reabsorption of sodium ion, water should move after that due to homeostasis. The movement of solutes into peritubular capillaries decrease the solute concentration of the tubular fluid, but it increases the solute concentration in the peritubular capillary. Here, if you have one tubule here, and near the tubule you have a blood vessel like this, when you have reabsorption of sodium ion from here is sodium ion. When we have reabsorption of sodium ion from the tubule to the interstitial fluid, and after that the blood vessel, the concentration of sodium ion in the nephron tubule decrease, and the concentration of sodium ion in the interstitial fluid increase. So the water should move after sodium ion to dilute this ion in the interstitial fluid. We call it osmosis, and this is the obligatory water reabsorption. Water can follow the solutes which become reabsorbed, and about 90% of water reabsorption take place by this obligatory water reabsorption. The other type of water reabsorption is called facultative water reabsorption, and it's regulated by ADH, the reabsorption of water which happens by the function of the hormones. When we become dehydrated, the ADH is secreted. This ADH can sit on the distal tubule cells and collecting duct cells and help reabsorption of more and more water. We call it facultative water reabsorption. In this slide, you can see the different destiny for one material. Sometimes we have substance A and only we have filtration for this substance. All the amount of this substance A which become filtered can be seen in the urine. We don't have any reabsorption or any secretion here. The other material is called substance B. For substance B, we have a filtration, but after filtration, partial reabsorption takes place. So the amount of substance B in the urine is less than this amount in the Bowman's capsule. The other material is called substance C. We have filtration of this substance. All of these substance C become filtered and we cannot see any of this substance C in the urine. And the other material is like substance D. We have filtration. Besides filtration, we have secretion of this material from the blood vessels to the renal tubules. So the amount of this substance in the urine is much more than this amount in the Bowman's capsule. So we have different material with different destiny in our kidney tube. When the filtrate start to pass in the nephron, most part of this filtrate become reabsorbed from this isotonic solution back into the body's circulation. This process of reabsorption is selective and it's quantitatively large. Reabsorption is variable and totally based on the needs of our body. In this slide, you can see the reabsorption in the proximal convoluted tubule. The glucose, amino acid, and sodium will be pumped out of the tubule by active transport. For reabsorption of this material, we need ATP. Sodium is positive. So after one positive ion becomes reabsorbed, due to homeostasis of charges, we need to reabsorb chloride. So chloride with negative charge will follow the sodium into the peritubular space, as you see here. After we have reabsorption of glucose, amino acid, sodium, and chloride, because the amount of this material increase in the interstitial fluid, water should move after them. So here we have water reabsorption by osmosis. Some compounds are present in high concentration in the filtered but low in the blood can move by diffusion. And the most important ion which become reabsorbed is our sodium ion. Normally, we should not have any glucose, amino acid, vitamins, and lactic acid in our urine. So all of them become reabsorbed. In the proximal convoluted tubule, we have one symporter which we call it sodium glucose symporter. By function of this symporter, two sodium ion and a molecule of glucose attached to this symporter protein can move and they can become reabsorbed. Also, the sodium ion can be carried by one phosphate and sulfate ion in one direction too. And we have one antiporter in proximal convoluted tubule, which we call sodium hydrogen antiporter. It can carry filtered sodium into proximal convoluted tubule cells, while hydrogen ion is moved from the cell into the tubular lumen. As the result of this exchange, sodium is reabsorbed in the peritubular blood and hydrogen is secreted into the tubular fluid. And here we have a protein which we call it aquaporin 1. Aqua means water, and the aquaporin is the protein, membrane protein which is permeable to water and help water reabsorption after reabsorption of sodium ion. Solute reabsorption in proximal convoluted tubules promote osmosis of water. Each reabsorbed solute increases the osmotic pressure first inside the cell tubular cell, and after that in the interstitial fluid, and finally in the blood. Water thus moves rapidly from the tubular fluid to the cells, and after that to the interstitial fluid, and after that it goes to the bloodstream to make osmotic balance in all parts of our body. In other words, reabsorption of solutes create an osmotic gradient which promote the reabsorption of water. As water leaves the tubular fluid, the concentration of some solutes like chloride, potassium, calcium, magnesium, and urea increase. So by diffusion, they can come to the tubular cells, and after that again by the process of diffusion, they can go to interstitial fluid and peritubular capillary. You need to know that reabsorption of this material have two different ways. Sometimes they can move paracellular, and sometimes they can do diffusion transcellular. The reabsorption of protein in proximal convoluted tube happen in proximal convoluted tube. The proteins are very small in their amount in the filtrate. So normally we should not have protein in the Bowman's capsule. And if we have any protein, first we have a process which we call it vesicular transport or endocytosis. By this endocytosis, our protein can go to the epithelial cells. When it enters the epithelial cell, inside the cell, it's broken down by different enzymes. So hydrolysis of them happen, and we have amino acid. The amino acids are very small, and then by the passive transport they can move from the basolateral border of the cell to the interstitial fluid, and after that to our blood stream. After the proximal convoluted tubule, we have loop of Henle. The chemical position of the tubular fluid in the loop of Henle is completely different from the proximal convoluted tubule because most part of the ingredient are reabsorbed in the proximal convoluted tubules. The glucose, amino acid, and other nutrients, and about 65% of filtered water are reabsorbed as filtered fluid move through the proximal convoluted tubule. As a result, the chemical composition of tubular fluid entering the next part of the nephron, which we call it loop of Henle, is quite different from that which we have in the glomerular capsule and the proximal convoluted tubule. When the filtered substance enters the nephron loop, we have reabsorption of about 15% of water, and we have reabsorption of sodium ion, chloride ion, and also potassium ion in the loop of Henle. In this picture, you can see the loop of Henle or nephron loop. In the apical border of the cells, in the thick segment of this nephron loop, we have a protein which we call it sodium potassium chloride symporter. This symporter help reabsorption of one sodium ion and one potassium ion. Sodium and potassium, both of them are positive ion. So after this positive ion reabsorption, we need to have two chloride, which is negative. So two chloride ion become reabsorbed. They come into the cells. The sodium ion can move to the interstitial fluid by the function of sodium potassium ATPase. So it utilizes one ATP and it becomes reabsorbed. The chloride ion can do diffusion from inside the cell to the interstitial fluid and after that to the blood vessel, and the potassium ion by leaky channels return back to the tubular filtrate. and again in the next step, in the next process, again it can come inside the cell. So this is the happening which we have in the loop of nephron. And you need to know that, sorry, I forget to tell you this. Although about 15% of filtered water is reabsorbed in the descending limb of the nephron loop, little or no water is reabsorbed in the ascending limb. In this segment of the tubule, the apical membrane are impermeable to water because ions, but not water molecule, and become reabsorbed here. The next part is the distal convoluted tubule. Cells of the distal convoluted tubule reabsorb about 10 to 15% of filtered water with sodium ion, chloride, and calcium ion. For the calcium ion reabsorption, we have some receptors in the distal convoluted tubule cells. This receptor is the receptor of parathyroid hormone, or PTH. When the PTH is secreted, it can sit on its own receptor and help reabsorption of calcium ion. You need to know that when this hormone is secreted, it inhibits phosphate reabsorption in the proximal convoluted tubule, and in the opposite way, it enhances the secretion and excretion of phosphate from the urine and from our body. The last part of distal convoluted tubule and the collecting duct have two different types of cells. You know that we have principal cells and intercalated cells. The intercalated cells are very important for maintaining the homeostasis of pH, and the principal cells are important for homeostasis of water. The principal cell becomes stimulated by ADH. When the ADH, or antidiuretic hormone, is secreted at the time of hypotension or dehydration, it can sit on the principal cell. It can active and stimulate the aquaporin. So this protein can do reabsorption of water. And after that, our sodium ion can become reabsorbed, and sodium potassium pump work here, and we have reabsorption of sodium and secretion of chloride. And the intercalated cells can do reabsorb potassium and bicarbonate. You know that the carbonate is very important for buffering system of our blood and also interstitial fluid, so we need it in different parts of our body. And it can secrete hydrogen ion when our blood becomes acidic. The homeostatic regulation of tubular reabsorption and secretion can be done by 5 different hormones in our body. One of them is called angiotensin 2. I talked about angiotensin 2 in the blood vessel chapter. One of them is aldosterone. The other one is ADH, ANP, and PTH. These hormones can regulate the reabsorption of sodium, chloride, calcium, water, and they can help secretion of potassium ion. Here you can see the pathway which we call it renin-angiotensin-aldosterone system. When we have hypotension, from our kidney we have secretion of renin. Renin can work on the angiotensinogen from the liver and change this angiotensinogen to angiotensin 1. Angiotensin 1, with the help of angiotensin converting enzyme, which is secreted from the lung, can change to angiotensin 2. Angiotensin 2 have different effects. First, it can do vasoconstriction and increase the blood pressure. Then it can increase aldosterone secretion from adrenal gland. By secretion of aldosterone, you know that we have a reabsorption of sodium ion from the kidney tubule. By reabsorption of sodium ion, water should become reabsorbed after that, so the volume of the blood increase and we have hypertension. This angiotensin 2 can increase ADH secretion. ADH is secreted from the posterior pituitary gland, and it can work on the principal cells uh, in our collecting duct, and it can promote reabsorption of water and sodium ion. And also, angiotensin 2 can increase thirst. So we drink water and the volume of blood increase. ADH is released from posterior pituitary gland. It acts on the principal cell of distal convoluted tubule and the collecting duct. It can increase the permeability of tubules to water by secretion of ADH. Vesicles which contain aquaporin 2 insert this protein to the apical border of the cells so they can facilitate reabsorption of water as much as they can. And the next hormone, which is very important here, is called atrial natriuretic peptide. When we have a large increase in the blood volume, the blood which return back to the right and left atria increase. So the wall of our atria goes under pressure. And from this wall, we have release of AMP. We have release of this protein. Atrial natriuretic peptide inhibits reabsorption of sodium ion and water in the proximal convoluted tubule and collecting duct. Atrial natriuretic peptide also suppress the secretion of aldosterone and ADH. These effects increase the excretion of sodium and water in the urine, which increase the urine output and decrease the blood volume and blood pressure. So up to now, ANP increase the amount of urine and decrease the volume of blood. Aldosterone and ADH are opposite. They increase the volume of blood and decrease the volume of urine. And the last hormone which works here is called parathyroid hormone. Although the hormones mentioned can work on the water loss in our urine, in our kidney tubules, but we have a hormone which can work specifically on calcium ion. When the amount of calcium in our blood decrease, the PTH is secreted. They can sit on the distal convoluted tubule cells and help reabsorption of calcium ion. When we have reabsorption of calcium ion, water can move after that. And you need to know that PTH is very important for secretion of phosphate into our urine. This part is complete. Next time I will talk about the male reproductive system.